Welcome to Part B of Chapter 8. This segment of video covers formally what is known as enzymatics. If enzymes can influence reactions by lowering activation energy, if they can speed up reactions, then by extension, if we change the activity of that enzyme, we can either slow down, modulate, or stop a reaction in a cell altogether. So we're going to talk about factors that influence enzyme activity in this part. A little bit of enzyme anatomy, if you will. One of the key landmarks of an enzyme is this area called the active site, where the substrate or substrates will bind in order to proceed with a reaction. That is a key critical part of the function of an enzyme, is that the substrate binds in an active site and is then handled or reacted. So if we have this active site, then how does the active site do its thing? Well, first of all, it can orient reactants correctly. It may torque bonds in order to strain them and cause them to break. Might provide a favorable microenvironment. We have all different kinds of side chains on amino acids. We have hydrophilic side chains, hydrophobic side chains, acidic, basic. And so we can create a microenvironment where that substrate wants to bind and react in a given reaction. We can also even covalently bond to a substrate and create a covalent intermediate. We'll show you some examples of this in the next couple of slides. Here's an example of orienting substrates. Maybe those molecules would be reactive, but they just don't bump into each other in the right way often enough, and so the reaction is quite slow. Well, the enzyme might be able to facilitate that by getting these molecules to orient properly. This right here, we have a polymer, and the enzyme is breaking that polymer into two smaller subunits. Perhaps in a way of straining that bond that holds them together, it makes that bond more sensitive to the ambient temperature and thus will break. Providing a favorable microenvironment, this one's a little tougher to find images on. Most of the ones that I've seen are usually pretty hard to interpret unless you have some experience. But here's an example. Let's say we have a substrate and it's dissolved in water. That microenvironment might actually be more favorable for the substrate than the aqueous environment. And so this is describing what we call a phase transfer, where the molecule is dissolved and now the molecule is out of the water and now is in a different environment, and maybe a reaction will occur. And lastly, this idea of covalently bonding. This is a transient covalent bond, like for instance the enzyme and the substrate here. It'll form a sort of a covalent intermediate as electrons are shifted around, and then once that one reactant is gone, the other one will release. It's a way of holding on to electrons transiently before releasing one or more of the products. Figure from your book kind of showing this, I would kind of consider this, orientation, or perhaps a microenvironment where now these substrates can actually react, and so we have this binding event. When they say weak interactions here, they presume non-covalent. That means hydrogen bonding, ionic bonding, or hydrophobic interactions. And so we create this complex. That's what we mean by the enzyme substrate complex. The environment is created, the orientation is achieved, we get a reaction, and now these products have changed somehow. In this case, we're not fusing the two substrates, we're doing some other type of a reaction, and so we have two products that are released. And then we can recycle this enzyme, and that's why we call it a catalyst, because the enzyme itself is not changed. We're talking about enzymes, but I want you to know that the same principles of enzymes apply to carrier proteins. You have a binding, change, and conformation release. You have a binding site, which is a lot like an active site. And so carriers behave just the same way that enzymes do. In fact, you could say that this carrier protein is lowering the activation energy requirement for molecules to move across the membrane. And so an enzyme is a complicated three-dimensional structure that has a given activity. If the structure is ideal, then the activity is at its maximal level. The activity is the ability of the enzyme to perform a given reaction or function. If we change the structure in any way, the odds are that it will also affect activity of that enzyme. And so an enzyme's activity can be affected by things like pH and temperature, environmental factors, ionic concentration. All of those environmental factors have the potential to influence chemical bonding. So if we raise the temperature, weaker bonds might be broken, or if we lower the temperature, it might form spontaneous bonds that normally wouldn't form. Same thing with pH, though we're more specific here. We're talking about hydrogen bonds. Too few pH molecules, that would be a high pH, would mean that maybe some amino acids form hydrogen bonds that normally wouldn't. Too many protons, too low of a pH, maybe those protons are interfering with hydrogen bonds. And remember that hydrogen bonds are at the secondary structure, the tertiary structure, and the quaternary structure. So it's a major influential factor here. We can talk about chemicals that specifically influence the enzyme. We're talking about cofactors and inhibitors. Cofactors generally increase activity of an enzyme or allow activity to occur. Inhibitors are reducing activity or preventing activity. And we have two flavors of those, competitive and non-competitive. But we'll talk about those a little bit later. Let's start with the environmental factors, and that includes temperature and pH and ionic. We don't really talk about it here, but ionic, if we have ionic bonds, then ions can influence this. Temperature, pH, we're talking in ions, we're all talking about the potential for modulating or interfering with chemical bonds in the three-dimensional structure. And that's why we would have an optimal temperature that an enzyme has maximal activity. If we deviate from that temperature, the activity changes. Same thing with pH, though what we're dealing with here is hydrogen bonds. Too many protons, a low pH, would interfere. Too few protons, and now we have maybe accidental hydrogen bonds forming. Maybe ionic as well. So optimal conditions favor the highest activity for that enzyme. So we get the proper three-dimensional structure, we're going to have activity. But if we start altering those bonds, those hydrophobic bonds, those ionics, those hydrogens, and in extreme cases, covalent bonds. For instance, if someone has their hair straightened, what they're doing is they're exposing their hair to chemicals which will break the disulfide bonds which hold their hair in a curly state. So optimal conditions include pH, temperature, and ionic concentration for appropriate bonding and appropriate shape of the protein. This is what I consider the crown jewel of Yellowstone National Park. It's called Grand Prismatic Spring. And the colors that are achieved here are a combination of the water and the organisms that live in that water. It's very, very hot in the center, so we don't have very much life there, but these extremophile bacteria are what give the colors that we see when it starts to be green and then yellow and then orange and then even this brownish color. Those are different kinds of bacteria that are thriving in different microenvironments in this spring. The ones that live near the center of the spring where it is hottest and probably most acidic as well have proteins that are adapted for that condition. Hardly anything lives in the middle of this. It's just too hot. It breaks bonds too easily, too acidic. But on the edges here, now we have conditions where proteins can be created that allow these bacteria to survive and even thrive. And that's what we're showing here. For instance, we might have two enzymes, this black one and this red one, that do the exact same reaction, that do the exact same thing, only this red one does it in heat-tolerant bacteria that we find in that spring in Yellowstone. And this is the enzyme that we find in human beings, which runs at 98.6 degrees or 30 Fahrenheit or 37 degrees Celsius. You can see that the bonding and so forth are optimized for this 37-degree temperature. We get higher than that and activity plummets. We're breaking hydrogen bonds, we're breaking van der Waals forces and ionic bonds. We get too low, maybe we now don't have enough wiggle in the protein. It's not behaving the same way that it normally would. Maybe new bonds are forming and so forth, and now we can even drop off to zero activity. But at 37 degrees, we can get zero activity in this enzyme over here that is designed to operate, has bonding profiles that hold it in its proper shape at close to 200 degrees Fahrenheit, or somewhere around 77 degrees Celsius. So the bonding profiles can give an optimal temperature for basically identical functional enzymes in two different organisms. Same thing's true for pH, only this time both of these are human enzymes. One is called pepsin, which is a protease. It's an enzyme that breaks peptide bonds. Trypsin is another protease. It breaks peptide bonds as well. It's just that pepsin is designed to work in the stomach when the pH is very, very low, lots and lots of protons. So it can function well at the same pH as roughly lemon juice. We get lower than that, yeah, we start interfering with bonds. We get higher than that, we might start interfering with the structure. By the time we hit to pH 4, there's zero activity. Doing the same similar chemical reaction, this trypsin is set up for pH of 8. This is an enzyme that's secreted from the pancreas, which also happens to secrete an ion called bicarbonate that neutralizes stomach pH and allows trypsin to do its thing. It inactivates pepsin and turns on trypsin by modulating pH. There's an example of control right there. We modulate the pH and now we modulate enzyme activity. Now let's talk about the chemicals that can influence enzyme function. We're going to start with the helpers, those things that we call cofactors. Generally speaking, there are exceptions. These are non-protein molecules and atoms that help an enzyme create its proper shape. So for instance, here's a cofactor that is helping with the active site of this protein. So when it binds, it now creates an active site that the substrates can bind. It doesn't have to be in the active site. It can be a non-active site area, but it's quite common that it is in the active site. Cofactors don't even have to be organic molecules. They can be inorganic, like a metal ion. For instance, calcium is very, very common as a metal cofactor in cells. Magnesium is also, or if we think about hemoglobin, what's the metal that we think about hemoglobin? Iron. So iron in the hemoglobin helps create an active site for binding oxygen. So cofactors can be organic, they can be inorganic. Oftentimes, if it's organic, we'll call it a coenzyme. You ever heard of acetyl-CoA? Coenzyme A. It's an organic cofactor that's bound to an acetyl group. Coenzymes can also include vitamins. For instance, a derivative of vitamin A is a coenzyme for the photoreceptor in your retinas. So they can be all sorts of molecules. Even proteins can be cofactors. I get a little bit itchy when I read this phrase, but they can be organic molecules, inorganic molecules, vitamins, anything that helps the enzyme do its thing, cofactor. Then we have what we would say inhibitory cofactors, but we just call them inhibitors. We have two flavors of inhibitors. We have what are called competitive inhibitors that compete for the active site. They have affinity for the active site and they can compete. They might bind, or maybe the substrate binds. Two things I'd like you to write down here, or just make sure you make a note of it. When it comes to competitive inhibitors, affinity is important and abundance is important. Either the abundance of the inhibitor or the abundance of the substrate. I have a figure later on that I'll show you what I mean. Affinity implies potency. If you have something with a high affinity, it might outcompete the substrate and it's effective at a low concentration. If it has low affinity, maybe it'll bind the inhibitor, maybe it'll bind the substrate. So affinity and abundance. But hang on to those phrases. I'll show you abundance later on. Non-competitive inhibitors, on the other hand, bind something other than the active site. So here's a picture of a non-competitive inhibitor, and if you look at the shape here versus the shape here, this non-competitive inhibitor has changed the shape and has altered the binding kinetics to substrate. So it makes it harder for this to bind, or maybe impossible in some cases to bind, and so you've lowered the protein activity. Examples of inhibitors we see all the time. If you hear about toxins or you hear about poisons, pesticides, antibiotics, pharmaceuticals, most of our pharmaceuticals are in fact inhibitors of some kind. Aspirin and Advil both inhibit the same enzyme. It's just that Advil is a competitive inhibitor and aspirin is a non-competitive inhibitor. Here's that figure, but I just want to say that non-competitive inhibitors are sometimes called allosteric inhibitors. That is, they are binding an allosteric site. If we have an active site, we have an allosteric site, which can be anywhere else other than the active site. You could have an allosteric site over here or here. It just depends on the interaction that we're talking about. And this is a graph showing enzyme activity. In black here, so we have what's called V. That is the velocity of the reaction. How fast is a reaction occurring for a given amount of enzyme? Let me explain that for a second. If you have one firefighter fighting a fire, there's only so many buckets of water that he can throw on a fire. You have 200 firefighters fighting, they can throw a lot more buckets of water. And so you have to define per unit when it comes to velocity. But let's say it's one copy of the enzyme in this picture. So we have the enzyme here and we have no substrate, but as we add substrate on the x-axis, the activity goes up. Goes up and up and up and up, and then finally it hits this plateau. That's what we call saturation for a given amount of enzyme. We've hit what's called the maximum velocity, the maximum rate of catalysis for the amount of enzyme that you have. Okay, normal kinetics there. If we add competitive inhibitor, remember what I said about abundance? Okay, well, you see that. Let's go to the 50% mark. This is 50% activity right here. It's called the Michaelis-Menten constant. Don't worry about that. Okay. That's about the amount of substrate that hits 50%. Well, you can see that the same amount of substrate, we're not hitting 50% in that competitive inhibitor. But if we keep adding more substrate, we can eventually swamp out that competitive inhibitor, and we eventually get to Vmax. It just takes a lot more substrate than we had for the normal enzyme. That's where abundance comes in. We need more abundance of substrate to overcome that competitive inhibition. But with non-competitive allosteric inhibition, we've changed the activity of the protein. We've altered the Vmax. So it doesn't matter how much abundance we have of the substrate, we have a different Km. So Km is the amount of substrate required to reach half activity. We've altered that in non-competitive. It doesn't matter how much substrate we have. With competitive inhibitors, abundance matters, along with affinity. Enzymes can and do change over time. They can change over the time of one's lifespan, and they can change from generation to generation through heritable and inherited mutations. So the central dogma says that we have this heritable material in the form of genes in our DNA, and when genes are activated or otherwise turned on, we transcribe them into mRNA, and then that mRNA is translated into proteins using ribosomes. That's the central dogma. But if we change the DNA, we will change the sequence in the RNA, and we could potentially change the protein. That's the basis of things like cancer, but it's also the basis of other diseases in the body. Mutations can and do lead to changes in the amino acid composition of an enzyme. Most of the time they're silent. Occasionally we'll get one and it's bad, but every so in a rare while, maybe a mutation's also good. Let me show you what I mean. These guys right here are the champions in the Tour de France, the big bicycle race that goes on usually in the summertime. The overall winner is usually the best all-around cyclist in the whole thousands of miles of race. So he's a good general rider. It's a general category. This guy in the polka dot jersey over here is what they call the king of the mountains. So he wins all of the big climbs. He's got points for getting all these big climbs. And then the guy in the green over here, he's a sprinter. They're all great bicyclists, but they're all great bicyclists in different ways. This guy over here has an isoform of myosin that allows him to generate huge amounts of power when the time is right. He's probably terrible in the mountains. He doesn't have very good endurance. This guy's probably terrible in the sprints, but he's good for cranking away on a big uphill. And this guy has probably a little bit of both. He can turn it on when he needs to and not. That's almost certainly related to the forms of enzymes that they have in their muscles. A guy with endurance may have very high activity lactate dehydrogenase. A guy with power has an isoform of myosin that can generate power quicker. That's oftentimes what makes the difference between a really great athlete and somebody who is just an average athlete. But enzymes are encoded by the heritable material in the DNA, and if there are any changes that aren't repaired, that can lead to amino acid changes. And I said most of the time they're silent. These changes occur in non-coding DNA. But if it happens in the coding part of the DNA that we make mRNA out of, maybe it keeps the amino acid the same. There is some redundancy there. But if it's in the right place, it'll change that amino acid and change the enzyme. Altering that enzyme may alter its structure and its ability to bind substrate. Every once in a while, you get a different mutation that confers a benefit, and I'm going to show you some of that. I'm going to show you examples of that. This is the basis of evolution when it comes to molecular evolution. The classic case in point, antibiotic resistance, a huge problem in the modern world because we aren't regulating antibiotics well enough. So people are taking antibiotics all the time, and guess what? They pee them out into our sewers. Well, there are bacteria out in those sewers, and a lot of them probably aren't resistant to antibiotics, but maybe one has a mutation that makes it resistant, maybe 1 in a billion. Well, if that bacteria are being killed off by the low levels of antibiotic, maybe those ones that have that one mutation survive and can reproduce and keep reproducing and keep reproducing. This is one of the reasons why we're beginning to develop organisms that are completely resistant to all known antibiotics. And it's a real problem because it's really hard to develop new antibiotics that aren't also toxic to ourselves. This is one of the coolest videos that has come out in recent years, showing this idea of evolution and literally in real time. So what you have is you have these bacterial colonies, and you've got tons of bacteria over here, and then you have a zone of antibiotics. So they took this colony and they put it right up against the antibiotic, and only a few colonies, only a few cells can survive, and you can see them grow and reproduce into this antibiotic. So they have resistance. They have an enzyme that allows them to resist that antibiotic. But that's not the end of the story. We hit this line right here, it's right here as well, and then you can see one colony really go off because it's not only resistant to this one, the enzyme that it has is also resistant to this new antibiotic. So we're literally seeing evolution in real time. Most of these guys didn't survive. Most of these guys don't survive the second insult. So whatever makes it into this next zone is going to be the one that's going to be most likely to reproduce and thrive in the new environment. That is literally evolution in real time. We're going to stop right there for part B, and we will pick up with a few more slides for part 3, part C. Thanks for your time, and as always, if you have questions, just let me know. Talk to you soon.